Okay, in part three of our four-part series, we're going to talk about some of the marine life in the estuaries. First, going to identify and discuss bioindicators of estuarine health. Going to then move on to biodiversity, term I'm sure you all have heard. Going to follow that up with brief description of food webs compared with food chains. Then I'm going to talk about the base of the food chain, the phytoplankton, followed by a description of some of the more common zooplankton or zooplankton. And we're going to finish up with a brief discussion of oysters, shrimp, crabs, and finfish. Okay, historically and even today, we use benthic biomass, its abundance, and species diversity as indicators or bioindicators to signify the health of the ecosystem. See some of the typical infauna or organisms that are found either in or on the sediment. They are used to indicate long-term health of a particular ecosystem, estuaries especially, because they really don't move much. If the water quality above them gets poor, they can only quite often move no more than an inch or two a day, whereas actively swimming organisms, fish, crabs, shrimp, if the water quality gets bad, for whatever reason, low dissolved oxygen, some kind of pollutant, they'll actually move to a more suitable area. These guys can't move, so what we do to look at the health of an estuary, for example, is go back and take a mud grab, say monthly or every 6 months or even every year from the same location, and sieve the organisms out of that mud. Count not only the numbers of organisms, but the identify the different species. And of course, what indicates a healthy ecosystem is not so much the sheer number of a particular organism. In fact, that quite often is an indication of pollution. But what we want to do is find a variety of organisms in that ecosystem. If we find that biodiversity decreases over time, we can pretty much be assured that the water quality over time has been degraded in that particular area. Again, as we just mentioned in the previous slide, that the benthos are often used as bioindicators because they, again, occur throughout the estuary, can travel only short distances if they want to avoid degraded water quality. They're generally osmoconformers, which means that the salinity may change, but it's not necessarily going to be something that's detrimental to their survival, which is good, because what we want to find is are there factors in the estuary other than the ever-changing salinity that is negatively affecting their survival. Again, we don't use fish, crabs, or shrimp, for example, because they can move and will move to avoid stressful conditions, including rapid changes in salinity with, say, a heavy rainfall or prolonged drought. So if we're going to utilize biodiversity as an indicator of the water quality in an estuary, for example, here's a good example of what you would typically expect in a healthy estuary. Number of freshwater species in the head of the estuary at very low salinity is going to be relatively high. Number of species here, salinity down here. You can see that then you have an area here from just right after freshwater up to about maybe 20 practical salinity units or parts per thousand, where you have true estuarine species. These are species that you typically find in an estuary because they can tolerate a wide range of salinity. And then, of course, from about 5 PSU up to 35, you have a true marine species. With, of course, as the salinity increases, the number of true marine species increases as well. So fluctuating salinities do limit the number of both plant and animal species, not the abundance, but the number of species to those which are truly euryhaline, or true estuarine species you see here in the green, versus those species that are strictly freshwater or tolerant of salt water. These would be species that are referred to as stenohaline. Spartina, for example, is euryhaline. We discussed it a little earlier. That's that fringing salt marsh plant that actually survives by excreting salt onto its blades. Some of these other organisms that you typically associate with saltwater, stingrays, bull shark, tilapia, killifish, can actually all tolerate fresh water for prolonged periods of time. So they would be euryhaline as well. Okay, this is somewhat of a complex slide, but it's got some really good information on it that we're going to try to go through without spending too much time. Again, freshwater inflow and salinity gradients that are created in our estuaries determine pretty much both the plant and organism distribution within the estuary. Again, as we just saw, there are true estuarine organisms which can tolerate a wide range of salinities, but again, most of them, plant or animal, do have optimal salinity range where they are least stressed osmotically and most likely to survive within that estuary. So you'll see zonation, say, going up onto the shoreline. You'll see, as I mentioned yesterday, the seagrasses growing in the clearer part of the estuary closer to the Gulf of Mexico. Again, estuarine trophic structures, that feeding trophic structures are some of the most complex in nature. And again, most are significantly influenced by salinity. And just quickly to look down here, you see that you've got a typical estuary here with a salt marsh on one side, which would be typical of Texas estuaries. And on the other side, this wouldn't be the same estuary necessarily. You've got, say, what you find on the west coast of Florida with a lot of mangroves taking the place of the salt marsh estuary vegetation that we have here in Texas. And then, of course, here, this is the estuary itself. Beneath each one of these, it's interesting if you take some extra time to look. You're going to see down here at the bottom the producers in that particular part of the estuary. In this case, it's salt marsh plants. In the water, it's either phytoplankton or diatoms and seaweeds. Over here, of course, would be the mangroves. All of these, the mangroves, diatoms, and the plants, contribute, as I mentioned the other day, to what's called the detritus or the detrital food chain. As they die and begin to decompose, they form detritus, which then you can see feeds deposit feeders, feeds some herbivores, deposit feeders on this side, even some filter feeders, and this detritus contributes both to deposit and filter feeders within the estuary itself. So a very, very important link in estuarine food chains is detritus. So you can see here, if you just follow the shore-based plants, you've got the salt marsh plants like the Spartina we mentioned, got herbivores that are feeding on that, as well as insects. Carnivores are feeding on those herbivores. Over here you can see that You've got phytoplankton that are contributing to oysters, for example, burrowing polychaetes, a variety of filter feeding organisms within the water column itself. So anyway, we could spend almost all day on this, but just to give you some idea of what to look for here, you can see over here you've got these are color coded, so you got herbivores with the green arrows, you got predators with the purple, you got detritus feeding export like here with the white arrows, and then you've got the blue showing contributions to the detritus pool. Over here, that's partially blocked by my video, is just a representation of all of the interconnected organisms, plants and animals in an estuary in Australia. And it's I just threw it in there just to show you how extensive some of these estuarine food webs can become. It's just a slide I want to show real quick to make sure that you don't get confused by the terms food web and food chain. There aren't that many food, well, a food chain basically is a kind of a single organism or group of organisms being fed upon by the next trophic level, which then is fed on by the next trophic level, and so on up. That would be a chain, literally link, 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 link. What typically is more likely in the environment, especially in estuaries, is a food web. And here you see basically the same representation that you had here as a food chain, but a food web, showing that there are a variety of organisms, in this case energy from the sun, producing diatoms as well as dinoflagellates that are being fed upon, instead of over here just by copepods as a primary consumer, by shrimp, copepod, pteropods, these in turn being fed upon by filter feeding fish, and these fed upon by larger fish. So basically, again, food chain, cartoony, one fish eats the smaller fish, eats the plants, whereas more likely what you find is a food web where there are numerous opportunities to feed on different size and types of organisms depending on the next level of the food web or food chain. Okay, in the estuaries, a large part of the primary productivity or the plant production actually occurs in the water column itself, carried out by microscopic organisms. In the open ocean, actually, the entire food chain is dependent on microscopic phytoplankton or microscopic algae to initiate the food chain through photosynthesis utilizing sunlight. Well, what you want to recall is the reason that this aquatic system is so different from a terrestrial system is the fact that water is not as solid or stable as land, and so in order for the plants that start the food chain in the ocean to remain within what we refer to as the photic zone, the top typically 50 to 60 feet, in real clear water maybe down to a couple hundred feet, where they get sufficient sunlight to photosynthesize. So that's the big difference. So in the ocean, everything starts out microscopic. On land, not so much. Again, the phytoplankton, that is the plant plankton, also referred to as algae, transfer carbon dioxide from the ocean to living matter. Algae are plant-like, but they're not really referred to as plants like land plants because they don't have vascular water and nutrient systems. They're so tiny, basically. Everything occurs as far as metabolism by diffusion in and out of the plant. So instead of being classified as plants, even though they're photosynthesizing, they're classified as protists. Again, they fix carbon, nitrogen, and phosphorus from carbon dioxide, inorganic nitrogen, and phosphorus, just like land plants. And most of that's returned to the near surface waters when these plankton are very rapidly consumed by the zooplankton, which are also microscopic in size, largely. So they're the next part of the food chain. So the zooplankton are feeding on the phytoplankton. They're excreting little fecal pellets that are broken up and dissolved, releasing nitrogen or phosphorus for more photosynthesis, as well as bacterial decomposition of both dying and dead algae and zooplankton. We'll see on the next slide, but just to introduce it here, that some of the oceanic algae and zooplankton actually sink out of the photic zone before they're being eaten or decomposed, unlike in our estuaries that are relatively shallow. The nutrients then released into the deeper waters, lacking sunlight, and don't get utilized until they are brought back to the surface through a process referred to as ocean upwelling. By comparison, again, if you look here at land-based systems, you see some trees growing here in the forest. You see this forest litter. Actually, when these trees or when the trees lose their leaves or when the grass dies, the nutrients pretty much get washed right back into the root zone to be retaken up by that same plant. So it's a system that, because of the fact that land is more dense, more solid, if you will, than water, you don't have these nutrients potentially sinking out of the sunlit zone, but rather they're being recycled right within the root system before they can percolate down to deeper water and eventually get into aquifers, for example. I want to move offshore for just a minute and talk about open ocean algae production. If you notice here on this world map, the yellow areas have the most abundant algae production, and that's because the nutrients that we've been talking about along the edges of the continents here are washing in from the surface, land surface through the estuaries and into the near shore open ocean, where they're stimulating the algae to grow and start the basic food chain. When you look out here in the deeper ocean that we used to consider a biological desert, you can see that there are nutrients that are coming up from the deeper water. If you look here, you can see that out in the open ocean, unlike our very shallow bays, when you get down to about from the surface to 50 feet, there's hardly any nutrients. They've all been used up closer to shore. But unlike our estuaries that are pretty well mixed from top to bottom, in the case of the open ocean, you find the nitrogen and phosphorus in particular increasing very rapidly down to about a 1 kilometer depth. Both nitrogen and phosphorus kind of got a little hump here, and then it levels off. But see, it's about 300 times more than you would have at the surface. And when this deep nutrient-rich water moves back up close to the surface through a process called upwelling, which is wind-driven circulation, you get areas like right here along the equator where this deeper water is being brought back up to the surface, stimulating phytoplankton production and ultimately an entire food web that results in some very important commercial fisheries. For example, most of the tuna fish that you eat came from this area along here. Another example down here where particular wind-derived systems result in what's called gyres. You can see that, again, some of this deep, nutrient-rich water is being brought up near the surface, and this satellite photo, again, shows abundant algae production here. On the up here near the North Pole and the South Pole, where the filter-feeding whales, the baleen whales, head for their feeding in the summertime up here, in the wintertime down here, because that's when you have almost 24 hours of sunlight on those respective poles. See, there's a lot of algae production up here, and that's basically coming not so much from, it's basically upwelling, but it's from very deep ocean circulation that's coming up near the surface as the bottom of the ocean gets shallower here. So you get this very nutrient-rich water coming up and stimulating these extremely dense algal blooms that ultimately result in a very productive food chain. So okay, I want to kind of rapidly go through our various phytoplankton that you find in estuaries as well as offshore that start the food webs in the ocean systems. I'm going to start off with the cyanobacteria, the blue-green algae. They're relatively small, again 1 to 700 microns. They include a recently discovered 0.6 of a micron genus Prochlorococcus, which is now referred to as a picoplankton, and is thought to be the most abundant life form in the sea. This organism is part of what's discovered back in the early 80s, part of the microbial loop, and which may provide over half the world's oxygen supply. Next, we're going to look at the diatoms. This is the most common primary producer or autotroph algae in the bays, because they prefer high nutrient levels that we find in our estuaries. They're fairly large, anywhere from 2 to 400 microns, or up to 4 millimeters. After that, we got the dinoflagellates that are varied. They, unlike the diatoms, the dinoflagellates are found in nutrient poor conditions. So not only can they photosynthesize, but they can also live heterotrophically if they don't have sufficient nitrogen and phosphorus to grow, surviving on dissolved organic matter as well as copepods, fish eggs. These are the green algae, or the zooxanthellae, that are found in coral reefs. And when a coral reef gets stressed, usually because of temperature or storms, they may expel these zooxanthellae, which normally are actively photosynthesizing and contributing to the health of the coral polyps, the animal coral polyps. They will expel them, and the reef becomes just white, like you would actually pour some bleach on it, hence the term bleaching. The red tide dinoflagellates are also responsible for the red tides that we get, which are toxic to marine life. You find red tides in Texas quite often originate just offshore in the fall, and may kill quite a few finfish that wash up on the beach. If they wash into the bays, the toxin that the red tides produce are also toxic to humans, and oysters feeding on the microscopic algae will concentrate them and could make consumption dangerous for human beings. So when that happens, the health department shuts down the oyster reefs until the red tide bloom is gone. If you've ever been out in a boat in Texas bays at night and you see a greenish white trail behind you, those are dinoflagellates lighting up, just like fireflies through luminescence. We don't have many green algae in the marine environment. We have some coccolithophores that are pretty common offshore. Particularly, they have calcium carbonate rather than silica shells. And one of the concerns we have with global warming, atmospheric warming, because of carbon dioxide, we think that carbon dioxide is soluble in seawater. It actually reacts with water to form carbonic acid, which lowers the pH, or increases the acidity of seawater. And we're concerned that perhaps the lowering the pH just a little bit of seawater, particularly out in the open ocean where you find most coccolithophores, could inhibit the formation of these calcium carbonate shells. This is just a aerial photograph of a red tide bloom. How it appears. I think this probably came from red tide bloom in Florida. They tend to get these blooms almost annually, sometimes more than once a year. Here in Texas, we typically only get a bloom, maybe every 4 or 5 years. This slide shows one of the more common zooplankton that you find in Texas estuaries, as well as offshore. When you're describing zooplankton, there's two major groups. There's plankton that stay microscopic pretty much, their whole life. These are called holoplankton. These copepods are an example of that. We'll see some other plankton in subsequent slides that are only plankton in the very early stages of their life, and they're called meroplankton. This particular copepod, Arcacia, has been observed in a laboratory to consume up to 300,000 diatoms in a day. They're pretty voracious, and of course, as we saw in an earlier slide, diatoms are one of the mainstays of the phytoplankton community in bays. The bottom slide down here you can see is a Nauplius copepod. It's a little bit smaller. It's basically what hatches out when the egg hatches. And since these are have exoskeletons, they have to molt in order to grow. So this Nauplius copepod at 1/10 of a millimeter will go through 6 molts. Nauplier molts, followed by 5 molts after it looks more like the adult copepod, in about 2 to 6 weeks. So that they're molting quite frequently. They live about 6 months to a year. I don't know if anybody's kept them in laboratories longer, but typically they are going to be eaten by something, usually within 6 months to a year. Most zooplankton are classified as holoplankton because they're microscopic in size, which is why they get moved around by the current because of their small size throughout their entire life stage. There's one good example that we have here in our Texas bays is as an example of a holoplankton that does not necessarily stay microscopic, and that would be our jellyfish, as well as our comb jellies. Even though these jellyfish worldwide, some of them can get up to 8 feet wide and have tentacles a couple hundred feet long. Most of them here in the Texas bays are 8 to 12 inches in diameter, with tentacles maybe a foot or 2 foot long. But they again are kind of the exception in being holoplankton, in that even as adults, they even though they can pulse and move around, they still are not considered to be meroplankton because they're not actively swimming against the current they're living in. They're primary predators on estuarine and zooplankton, and then of course they provide feed for a variety of other marine organisms, in particular the sea turtles love to eat jellyfish. Probably our best example of larval fish, our best example of meroplankton would be larval fish that, as we said before, hatch out between 1 and 20 millimeters in size, obviously so small that they can't actively swim against the current, so they're part of the plankton for maybe the first couple of months of their lives, and then as they continue to grow and get large enough to actively swim against the current, they then become no longer part of the meroplankton, but now they're referred to as nekton. Here's some other zooplankton types. There's the top picture here. You see some blue crab zoea larvae. These are the larvae that hatch out as the blue crab eggs develop. They're typically about a half to 1 millimeter. They're going to be hatching out in the nearshore Gulf, even along the beach in the early fall, and eventually they're going to metamorphose several times into a megalop that we'll see later that'll move back into the estuaries where they find a critical habitat for the rest of their life. Down here you see a barnacle. Obviously, when it's first hatched from one of thousands of eggs that are spawned at a time, it's only a half a millimeter. It'll go through, because again it has an exoskeleton, it'll go through about 6 molts to become a cyprid larvae, which then will seek a suitable hard surface, such as preferably any hard surface, and you can see the barnacle actually then attaches, stays there the rest of its life. Here's one closed up, and here's one with its upside, with its legs sticking out, catching plankton and filter feeding, which is the nature of barnacles. Here's a slide showing the life cycle of the eastern oyster, or our typical Gulf estuary bay oyster. These animals are quite prolific in producing eggs. They can spawn up to 100 million eggs in a spawn, and may spawn 2 or 3 times during the summer. The eggs, of course, are extremely small, 7/700ths of a millimeter, or 70 microns. And typically these oyster reefs will begin spawning when the water temperature gets up to about 25 degrees, 25 to 30 degrees, and the salinity is at least 10 parts per thousand. They'll go through, like a lot of other invertebrates, they'll go through a planktonic stage as a villager larvae, and then they will metamorphose into a larva with a foot called a petavillager. They'll then look for a solid surface to set down, similar to the barnacles that we saw in the earlier slide. They'll set down a spat, and during their entire life from then on they'll stay in the same spot, and they'll feed primarily on plankton. They can also, of course, retain pathogenic bacteria and toxic dinoflagellates that we referred to earlier in the red tide. They play a critical role, oysters, in bay ecology by filtering up to 50 gallons of water a day. So if we've got a bay system that's receiving a lot of nutrients, becoming somewhat eutrophic, these oysters can can help knock down that excess phytoplankton population by consuming the phytoplankton as they're feeding and respiring simultaneously. They actually efficiently recycle bay nutrients through production of both feces as well as pseudofeces. It's kind of unique here. If there's an excess of algae in the water, but they need a certain amount of water to flow through their system just to respire, they'll take that excess algae and not even digest it. They will just accumulate it into little strands, and you'll quite often see what's referred to as pseudofeces. When you open up an oyster, you'll see the little strands of brownish material around the edges of the oyster. They mature at about 2 years for the female, 1 year for the male. Market size in Texas is 3 inches. It takes about 18 months to 2 years to reach 3 inches.